Great. Okay, so hi everyone. Thank you for the organization, and thanks Carlo for the invitation and giving me the chance to talk about my work today. I will be talking about some studies and a general kind of overview on how to combine cognitive training with TDCS in the context of aging. I will be talking about healthy aging because we always try to do the studies first on cognitively healthy population. I will present mainly data from behavioral and resting state acquisitions. To give an overview first, I will try to hopefully answer the question why it is important at all to elucidate the effects in older adults first, and then I will present some data from a study where we applied TDCS during a memory paradigm and investigated the effects on the behavior and their association with resting state functional connectivity. Then I will talk about a combined intervention of a multi-session cognitive training with simultaneous TDCS to improve performance on trained but also on other tasks. And I will end with an outlook and some open questions, and I'm looking forward to a discussion afterwards. Okay, so why is it important to elucidate effects in older adults at all? So TDCS has been shown to be a potential viable tool to counteract age-associated functional impairment. However, as age-related neuronal changes result in local changes in brain activity and disrupted connectivity between brain regions, stimulation of the same region may act differently in young and older adults. This is an example of how the same intervention may result in different results in a young and an older cohort. What has been done here is TDCS with the anode over the left inferior frontal gyrus and the cathode over the right supraorbital cortex during acquisition of a resting state fMRI. And here you see the results of an eigenvector centrality mapping analysis for a nodal versus sham condition in the same individuals for young adults, and here you see them for older adults. And mainly what I want to show here is that there is a difference. So in young adults, a nodal TDCS over the left inferior frontal gyrus mainly increased brain connectivity, so centrality in left and also contralateral right, prefrontal and posterior parietal regions, some temporal regions, and to a larger extent on the left hemisphere compared to the right, also because the simulation was designed to stimulate the left cortex, and in older adults we observed a different pattern. So there was an increase in posterior medial regions and a decrease in lateral regions, including the insula here in the frontal cortex. So these studies are important but still rare. They illustrated that the results obtained in TDCS studies with a young individual cannot automatically be translated to older populations. Due to substantial evidence of age-related network reorganization, a variability network reorganization, also an age-related structural changes among older adults, and also an age dependency of neural and behavioral effects of TDCS. So when stimulating aged brains, we need to make kind of a number of decisions. First, when defining the stimulation target, these altered activity patterns need to be taken account. So as task-specific brain activation patterns undergo distinct modification at advanced age, such as overactivation, under-recruitment, more widespread activation, a shift from occipital to frontal regions, to just name a few. We need to decide whether we want to promote these activations, which are thought to be to represent compensational mechanisms, or whether we want to restore a more youth-like activity pattern. Then we need to define where to place the electrodes to reach this target, and here not only the amount of global atrophy but also the location of the target region and its specific change caused by age-related volume reduction need to be considered. And the electrode montage can be based on predefined landmarks or also on individualized models of current distribution. Finally, we need to select the stimulation parameters to effectively modulate the target, such as the stimulation intensity, duration, and time window to induce the plasticity, selected according to standardized protocols or based on individual markers. Because in the end, the clinical value rests on the long-term effects requiring the targeted system to be sufficiently plastic. So now we want to be able to kind of make all these decisions in the context of cognitive aging through combining TDCS with cognitive training in older adults, to examine the general efficiency of a given protocol and its underlying mechanisms. We do single session studies, mainly in crossover designs, where we apply nodal and sham stimulation during a cognitive paradigm, mainly to understand the effects and the underlying mechanisms. And we also want to do multi-session designs that are mainly designed in a between group design, where we have a group receiving a nodal stimulation during a cognitive training and a group receiving sham stimulation, because here we think we may be able to restore functions or to produce some kind of sustained effects. At the same time, we want to, or we should, explore individual contributors to variability in responsiveness. These can be internal, related to the individual, or external, related to the protocol. And now I want to talk about a study, or present a study where we did the single session approach and acquired behavioral and connectivity data. So what we did here is we applied a memory paradigm where participants learned the association between words and pictures. It was a statistical principle where correct pairings were presented more often compared to incorrect pairings over the course of several learning blocks. And in the end, you can see that participants here in a baseline session with no stimulation are able to learn these associations. So starting at a chance level performance, going up to around 70%. And this learning success is tested in a subsequent retrieval sessions, where these pictures are replaced by a German word and paired again with the newly acquired word for this object. And also here participants are able to recall these words and to learn these associations. We then went on and applied TDCS in a crossover design over the left temporal parietal regions with 1 milliampere for 20 minutes, approximately covering the first 4 learning blocks. We applied a crossover design with a counterbalanced order. So the half of the subject started with sham and the other half started with a nodal. And you can see here that in sham, participants already improved compared to baseline. But when a nodal TDCS was applied, we observed steeper learning curves and also an increased retrieval performance after nodal TDCS. So we concluded that a nodal stimulation over the left temporal parietal cortex led to improved learning and memory performance in older adults. What we also did during the baseline session is we acquired resting state fMRI in the scanner, and we analyzed this data using a seed-based approach to quantify connectivity seeding from the left hippocampus, as this, this task and also this function has been shown previously to be mediated by the left hippocampus mainly. And what we found is that when doing seed-based whole brain analyses, that connectivity between the left hippocampus and the left angular gyrus here correlated positively with performance at baseline. So the higher the connectivity or coupling or synchronized activity at rest between the left hippocampus and the left angular gyrus, the better the performance before any intervention. And then the next step was to see whether this connectivity on an individual level also predicted response to the stimulation, and indeed the magnitude of TDCS response was also predicted by this functional connectivity between the left hippocampus and the left angular gyrus. And the most remarkably here, this was also the cortical target of a nodal stimulation, so giving us maybe an idea of the underlying mechanisms of this TDCS effects here. So to sum up the study, we showed that temporal TDCS can improve associative learning in older adults. We found an effect on learning and on retrieval performance. Resting state functional connectivity data acquired at baseline was associated with initial performance in the task and may be a predictive biomarker for behavioral TDCS effects. And this can help us to explain some inter-individual variability, maybe to distinguish responders from non-responders, and also to identify networks as stimulation targets. The next step, as I mentioned in the introduction, to restore or halt functions or halt the decline would be multi-session studies investigating the modulation of specific networks, the sustainability of effects, and also the transfer to tasks other than those which are trained. And this is what we did in another study where we combined multi-session cognitive training and TDCS to improve performance on trained and transfer tasks, and also to study the underlying neuronal network effects. So here we used a paradigm which the principle was similar to the paradigm I presented before, but here instead of pictures and words, the participants had to learn an association between objects, so buildings, and their actual place or a location on a kind of street map. This is a retrieval trial that is shown here in the learning blocks, similar to the word picture associations. The buildings were presented more often on their correct position compared to their incorrect position over the course of several blocks. And you also can see here, this is for young participants during sham, that participants were able to learn these associations also starting at the chance level. And you see the training gain during the first week, during the first day was highest, and then on the second and on the third day it was lower, reaching maybe some kind of ceiling performance. And here the main outcome was 1 day after this 3-day intervention in this retrieval phase. Here we stimulated over the right temporal parietal cortex with 1 milliamp for 20 minutes each day. And here I showed you the results for young adults with sham, and those young adults who received a nodal stimulation outperformed their peers receiving only sham. This was true mainly for the retrieval after the learning phase on each day, and also after the 1 day and the 1 day follow-up. Now, in older adults, you can see that we applied 2 more learning blocks. So they are able to reach a considerable performance level here. You can see that they also improve over the course of 1 day and also on the 2nd and the 3rd day, but they do not at all reach performance levels of the young adults. But when we then apply a nodal stimulation, we see that they are able to improve here, and then in the end they are better compared to those who were stimulated with a sham stimulation. So a nodal stimulation over the right temporal parietal cortex led to improved object location memory performance. We also tested performance on tasks other than the train task and found that 1 month after training, there was better learning and recall performance on the trained functions. So it was the same task, but another version of the task with other stimuli. And there was also a small effect on recall performance on a wordless learning task in the nodal compared to sham stimulation, the direction of better performance in the nodal compared to sham groups. Okay, so kind of the main aim here was to look at underlying neuronal effects. So before the intervention and also after the intervention, approximately 1 day before and 1 day afterwards, we applied again resting state functional MRI to assess the effects on functional networks. We here analyzed the default mode network, which is a central hub or central network with the precuneus as a central hub, mediating memory performance, being also sensitive to aging, and not only healthy aging, but also pathological aging, and also shown to mediate performance in this object location learning. And here we used ICA-based approach and also then extracted a default mode strength representing this integrity in this network. So, and what we see here is that the strength or the coupling in this network was increased in a nodal compared to sham stimulation. So the nodal stimulation led to increased coupling in this network, and this increase was also positively correlated with the performance increased. Obviously, we have only a few subjects here, but interestingly, this correlation was here mainly observed in older adults receiving a nodal stimulation, also giving us a tentative idea about possible underlying mechanisms of the effects of this combination. Okay, so in sum, we also found that temporal parietal TDCS can enhance the benefits of visuospatial cognitive training. We observed the effect on trained, but also on non-trained function. The combined intervention also led to increased functional coupling in the default mode network, and there was an association with performance benefits, mainly in older adults. Now the next steps, besides of course replicating these findings in larger samples with other protocols, but more on a clinical side, would be to perform randomized controlled clinical trials to study the efficiency of these interventions. And this was this kind of, I want to end by presenting our ongoing projects and the questions that are still open. So we are now conducting randomized clinical trials in older adults where participants perform cognitive trainings tailored to modulate executive functions. So after recruitment and telephone screenings, participants enter a baseline assessment where they're tested neuropsychologically and on the train task. And based on the performance on the main training task, they're then randomized into two groups, one receiving a nodal TDCS over the left prefrontal cortex and one receiving sham TDCS. Then going into a pre-assessment, performing all the two training and transfer tasks and MRI, and then finally going through the 3-week intervention where they perform the training with TDCS for 3 days each week, 9 sessions total, and then we have post-assessments and 2 follow-ups to study the maintenance of the effects. Just briefly, we have 2 training tasks, one focused on working memory updating, and one more of a decision-making task, where here they have to learn sequences of letters and recall the last 4 letters. And in the decision-making task, they basically have to learn sequences that produce a certain gain and those that produce monetary losses. And in transfer task, we kind of have designed the study such as there are some transfer tasks representing near transfer from the training and some that have more far transfer type. And these are ongoing studies, and we are also now performing all the analyses. Just briefly, some preliminary data. So, as I said, the data is still in progress, but what you can see here for the first training task is that here participants with TDCS seem to descriptively perform a little better compared to those that receive sham stimulation over the course of all the training blocks. But in the end, the statistical models that are appropriately adjusted do not show any difference in mean number of letters between the groups. And also here in the follow-up assessment, there is no difference in the TDCS compared to the sham groups. But what we observe is a minor effect on the near transfer task, which was the MBAC task, in the direction that participants that receive a nodal TDCS actually are better compared to those that receive sham stimulation during the intervention, and this effect seems to persist after 4 weeks, but not after an interval of 6 to 7 months. Interestingly, we have also an interaction in the direction that those individuals that have initially lower performance levels are also those that tend to improve from the intervention. Yes, so this in this study, but also in general, are the open questions still. So we want to combine cognitive training with TDCS in older adults, but we still don't know, is there an effect on the train task? Can we expect also the effect on train task? Is there specificities? So is the one train task improved and the other not? Is there an effect on non-train task? Is there a difference on some kind of transfer and none on the others? And also, do we have short or long-term effects? Can we expect some kind of sustainability? I can recommend some critical readings here. Also one very recent one, which summarizes also the challenges in the field. And in general, the evidence in the field is still very mixed. But I think it is important to also study the neuronal correlates of these training gates to maybe be able in the end to understand who actually responds and who would not respond do this intervention, maybe also explaining this kind of high inter-individual variability in the field. And also this would be important to identify network targets. So maybe we need other stimulation protocols also, as I showed in the beginning, to make all these decisions before we actually perform a study with TDCS and cognitive training in older adults. And the ultimate aim kind of is to judge the therapeutic potential of this intervention, for example, for patients with mild cognitive impairments, so the prodromal stage of Alzheimer's disease. So we are now also trying to go a step further into this direction, but for now we try to get an idea from the studies in healthy older adults. Yeah, and with this I want to end by thanking my colleagues and you for your attention, and I'm looking forward to the discussion.